Uh, okay, cool. So let's see here. So let me find, actually, the paper's not pulled up. So let me load Zotero again. This is actually the code. Um, so
Okay,
let me
now we can
see,
go.
get rid of that. Okay. All right, awesome. So the paper that I'm gonna go over is this GenePT paper. Um, so basically the the concept that the concept is basically to use the embedding GPT embedding. So it actually, so I haven't tried it yet, but presumably you could do this with any embedding. And so what they do conceptually, there's a good diagram here, is that first they just take, there's like a database of gene to description. So you can give it a gene, you get back a description of that gene. And so you just put that into the embedding model. They used ADA 2, but they you could do this with other ones, and in fact they did try it with some others, and then you get back a vector, and then first of all you can do certain clustering and other things just from there. But so there's also a technology called single cell sequencing, which means that they instead of, so normally what happens is you have a cluster of, you have a sample from a person or whatever animal or plant. You take that sample and then you like sort of just, conceptually speaking, you just stick it in a blender, find all the RNA, and then that gives you a, or DNA, depending on what you're doing. You can DNA or RNA, and it gives you like the expression level of the gene or the genes itself, so you can get for that whole sample, right? So there's different types of cells. If it's genes, then all the genes should be the same, unless you're looking at like certain epigenetic factors like chromatin and other things. Is it? I'm like, I don't know a lot about this. I'm just talking about my very high level understanding here. But in any event, or RNA, which is, so just to brief summary, genes are sort of like a program to make RNA. RNA, there's like a whole machinery in the cell that takes snippets of the DNA, and it uses that to replicate RNA, which then um, folds into proteins, and those proteins do various things. Some of them are so like proteins are just these long sequences of RNA that like spontaneously fold up into various shapes, and these sometimes these like perform functions in the cell, like building structures in the cell, or they can be sort of they can like sort of be have functions like the molecules can bind to other molecules and used for signaling, or they can actually bind to the DNA itself and cause it to amplify or suppress the expression of a gene. So there's like this super nonlinear, highly complex, sequence of things that interact with each other within the cell, and then also these proteins can be like sort of end up on the surface of the cell, and then other, when cells bump against it, or other things bump against it, then they can, you're signaling to those other things. So like that's okay, that's RJ's very, very poorly done genetics 101. So anyway, you have these cells. Um, so there's a technology now that has become common in the last couple of years, in which you can take each individual cell and look at the expression level of genes in that individual cell. So they just, sort of, you can imagine just like kind of like separating out the cells, and then, sort of like, sticking each cell individually in a blender and looking at counting the sort of incidence of RNA sequences in genes in that RNA expression of genes in that individual cell. Okay, so what we're looking at here. So first of all, does anyone want to ask a question? That was really, spontaneously bad explanation, but if anyone wants to understand that better, I'd be happy to do my best. Okay. Okay, good.
My quick feedback is A is a lot more intuitive than B. B looks like gene attention.
Yeah, so okay, so here, let me go over this diagram then. So what happens is you take one of these, so you have a cell, and then that cell it's gone through a set of cells, N cells, and they've gone through this sequencing thing. So you have a expression level for each gene for each cell, right? So that's what this matrix is. So then, and then what you do is you take the embeddings from each of from GPT or whatever, in this case GPT, and you just weight them by the expression level in that particular cell, right? So like I have all these genes. These are my embeddings for each gene. So like each row is a gene. And then I just weight that for each cell by that gene, and then I sum them up, right? So now I have a, for each cell, I have like a single embedding that corresponds to that cell, which is just the weighted sum of all of the embeddings for each gene, weighted by the expression level. Does that make sense? That's the core concept here. So I want to make sure that came across clearly.
Yeah,
Yep. Okay. And then there's a second mechanism that they also tried. So C is just another way of doing this, where they just even simpler. They just take they look at the 1,000 highest expressed genes, and then they just put their name into a string, and then it starts with like cell one, or in reality it's like a slightly longer prompt, and it just says like here's the here's like all the 1,000 most expressed genes in this cell, right? And then they stick each one of those into the embedding model, and out comes a sort of n embedding embeddings, yeah, n embeddings for all of those cells. Okay, is that clear, the mechanism? So this is like really simple in the sense that the high level mechanism is very straightforward. Of course, there is the really complex transformer underneath that is doing all the textual embedding, which is but something that many people in the paper club should understand pretty well.
cool.
Okay, good. So then like skipping over the related work. So basically, without going into too much detail, I think that I've been pretty clear about what they do. They have like these 30,000 genes. They do a bunch of deduplication. Because I mean, a big part of this, getting this to work, is actually just figuring out how to, like, all the different papers have different names for genes, and there's sort of like a whole bunch of databases, and they all use slightly different sort of stuff. So there is this one ensemble database, which is this right here. This ensemble database actually is like the goal of it is to take all the different names for a gene and just give it one ID. And so they use this to sort of unify everything. And then, okay, so then they have some different applications. They have this, sort of like, uh, um, so like functionality class prediction, meaning, so like what is the impact of, what is the function of this cell? And sort of like gene-gene interaction, uh, sort of like other unsupervised stuff, and then like looking at different cell states. And so they have data sets that are labeled or partly labeled for all of these things, so they can just use the embedding to test for, like, you test its accuracy and all these different things. Okay, so then, and then they have this, the results here. So if you look at You know, so they have this confusion. Okay, yeah, this confusion matrix, and you can see, you know, like it kind of gets a lot of stuff right. It looks like this protein coding one, which I think is one of the main things that you actually care about for RNA expression, is, you know, sort of has a lot of errors, and then this, sorry, like it thinks that a lot of things are protein coding when they're not, and it processing. I don't know what this process transcript means, but like that one gets wrong a lot. So like these, oops, so like you can see that like it seems to beat the competition pretty well. This gene former is um is a model that, you know, that's a little bit older, and then this SCGPT has gotten um a lot of attention recently. It's a also it's like a BERT, so similar to a BERT model. Or no, sorry, this one, GeneFormer is a BERT model. GPT is like a GPT model, and they you know do pretty good at, you know, sort of they beat these models. And I've actually used I'll show you guys, because we probably will run out of time. I've gone through the notebooks that the authors provided, which were pretty good. They're a little old, so they were broken, but I kind of fixed them. Um, and this was actually this was similar to the result I got. So these results do seem pretty valid. Um, but like, so maybe some of these other things are harder. Protein interaction, you know, so your ROC curve is like not so great. Um, so a lot of these other ones are, you know, but in any event, they're winning, right? So like this is, to me, I think this is like the headline is that it's just pretty incredible that you have these specialized models that were designed specifically to code for, you know, the relationship between genetic information, and that like instead we just take text that is a description of the gene that, you know, sort of briefly summarizes our knowledge about that gene, and then turn it into embedding, and it does pretty dramatically better than these specialized models. And I mean, I think if you think about it, it's not super surprising because you know, like the language has so much nuance, nuanced information in it, that if you use a good model, then you might be able to capture all that nuance better. Um, and but You know, like in any event, like it's pretty incredible how well they beat them. Um, and so this was like, so I didn't go through this part really in detail, but like they basically were trying to prove, okay, yeah, we're actually capturing underlying biology well. They're, you know, they sort of have these clusters of genes that make sense. Um,
By the way, I
and
think
then,
Kartik has a question.
Yeah. Hey,
okay,
hey, Ajay, sorry to interrupt.
no
One
worries.
question. So, could you like I didn't know if you had a chance to look at this, but like how exactly are they going from the vector representation of each gene to
Yeah.
a protein-protein interaction prediction? How does that exactly how do you go from that to predicting an interaction?
Um, Let me actually, that's a good segue here. I think that is this one. So if you look, so let's see, protein. So you asked about protein or gene?
I I, I mean honestly, either of them, whichever is simpler, I guess we can start with.
Yeah. So this one is the gene interaction. So they have these. Um, so Uh, let's see. Where's the meat of this? Yeah. So okay, right. What they do is they create a embedding of the two. They take the two gene embeddings and they sum them. Okay, so now you have like these two genes that um so you have two genes, you sum them together, and then you use that. So let's see, what do they do here? They fit, in this case, logistic regression. I actually tried also um light GBM, and it doesn't make a huge difference, but it's a lot faster. It does slightly beat the logistic regression, but it doesn't matter. Um, So let's see, they sum the two genes together.
Got it.
They
And I
do,
guess like so here like row of label is just like 0 or 1, I imagine, like for whether it's
yes,
interacting or not.
yeah,
Okay.
that's right. That's right.
Uh-huh.
Okay, does that make sense?
Yeah. So
Right.
okay,
So
so basically, yeah, you're training in logistic regression against the sum of the two genes and
yes,
trying to predict like yeah, whether they interact or not. Whether yeah, okay.
that's right. Yes.
Okay.
Is that
Yeah.
clear?
Yeah. I think that answers my question. Thank you.
Okay, awesome. Yeah, so it's actually, I was surprised that actually brings up a good point of how useful embeddings actually are, right? Like I wouldn't have, I think I had the same question in my mind, is like, so like, it's kind of obvious how to cluster genes and whatever, but I wouldn't have thought to do what they did to predict the gene-gene interaction. So I think it seems like there are lots of uses of these embeddings. Like there's been a lot of work, I think, previously using much less capable embeddings to do these kinds of analysis. So I think this is like sort of standard hat for bioinformatics, and then you just throw a much better embedding model and you get much better results. So yeah,
Yeah, I
and
think
they actually go
yeah,
please.
sorry. I think what's really interesting here is that like normally when people have tried to do this in the past, they try to assume like this like ground up approach where they either use the sequence itself or like some gene expression, protein expression, something, and they try to like sort of predict these interactions using that. Yeah, again, like very surprising that like just using text from the internet or like text
Mhm.
from any like standard description you can get like even like similar like and in this case it's like you're saying it's better, right? Like it's very surprising that you can get like better results. I think like yeah, one thing that might be happening I guess is that it's possible that a lot of those descriptions itself contain information about what other genes interact with the gene
Yep.
in question, right? So yeah, I guess that could be a confounding factor, but yeah, like across like millions of or I don't know I don't know what the size of the data set was, but like across even like tens of thousands of examples it would yeah it's really it's a really cool approach. I think.
Yeah. So I mean, I think you're exactly right. There is like information in those descriptions. I've looked at some of them about the interactions, and like so it'll say like this is, expressed commonly in cancer or whatever, right? So it says there's like so a lot of semantic content in those descriptions. And so that like it's not surprising after like in retrospect after they've kind of tried it, it's not surprising to me that it works. It's just kind of like it's pretty amazing how you like that it works so well to me. Yeah. And the other thing that this brings up is that okay, but what else can we stick in the description, right? Like you could I think they have left a lot on the table here on like okay, what if we start to put more complex descriptions that talk about like more explicitly about gene interactions, and like there's if you go to the whatchamacallit, the human genome browser and other things, there's like all this rich information about every every gene in the genome that we know about. And so that like what other information can you add to these descriptions to create like a really nice embedding that has like multiple purposes and so forth. So yeah, so let's see. It looks like they did so like um they did do some they like tried they say Llama 7B. I don't know what they did for this is not an embedding model, obviously. I don't unless they use some Llama 7B embedding. So they presumably are just using the class token as for the embedding. I don't know that it's because it hasn't been trained on contrastive text. It's not clear to me that that's actually a great comparison. Um, it maybe some of the, like something from the open MTEB, whatever that is, the massive text embedding leaderboard, right? Something from there might do better. So I was going to actually try that and see how it did. But interestingly, so while we're here, I actually did try, um, so like if you look here at this aorta data one, I actually tried, where is it here? Yeah, so I tried this also, the embedding 3 small, which is supposed to be a replacement for ADA. and, is pretty dramatically cheaper. But if you look at the results, so I did these plots, but I switched them back. Actually the performance was really poor comparatively, right? So that something about the ADA embedding, like presumably this was a distillation or something, and they lost probably scientific literature or gene codings, or I don't know what they lost in this, but it this model does way worse. So I found that to be quite interesting too. And so I definitely think that I'm going to spend some time and go through a bunch of models and just try to do the same metrics on a whole bunch of models and see what we can see about that. Because I don't think they did a great job in the paper of exploring embedding models, different embedding models. Um, Not to criticize the paper, it's an awesome paper, but that thing came short. And then, yeah, so then there's like, actually, this is what I'm reproducing. This line here is what I'm reproducing right now. And they have, this like SCGPT model, and then this is the two weighted and sentence versions of the thing. And you can see mostly like this, either SCGPT or this weighted model wins. Occasionally, it looks like for aorta, it actually, which like everything does kind of poorly on for phenotype, that this sentence wins for some reason, but not by a ton. So, okay, so let me see if there's anything else interesting. There was maybe one other table that I wanted to discuss briefly. Where is it? It's in the bottom here, I think. Yeah, here, this table here. So I found another thing that they tried was this ensemble of the three SCGPT plus the WNS model. And sometimes it looks like the ensemble doesn't hardly ever win. Like it wins these two, or no, I guess it wins 3 out of 6, so that's fine. But it looks like even when it loses, it tends to be close to the max, right? So that's not surprising for an ensemble model. So like the losing, like it lost here, but so it does seem like maybe it's worth doing an ensemble of different models. And the point that they make was in here is that like it seems like SCGPT is giving you like a slightly different, it's like complementary. The result is complementary here. So I thought that's basically all I did. They have some cool visualizations that are UMaps, but that's basically all I really looked at. Maybe I can stop here. I can go over my notebooks too, but they're pretty straightforward. So does anyone want to comment, ask questions, something? We're like half hour in, so plenty of time to talk, or we can go over something else entirely.
Well, just one question on this. Like, can we, apart from annotating, can we like generate
Well,
intro?
I mean, it's embedding, so no. But I feel that maybe, so I've been talking about this with my buddy that I mentioned in the beginning. Um, uh, the like, I think that there's a lot of opportunity here. So, for most papers, the great thing about medical research, biomedical research, is oftentimes that they publish the data. From, especially if it comes from an academic lab, you have a paper and you have a really nice, unique data set, right? So I think and I know that there are models that do this to some extent, where you like train on both the, sort of the gene or sequence level data and the text of the model. And then so that maybe like you get a model for which you can put in a cell and get a description back or something like that. Or the reverse, maybe put in a description and get out like a genome that matches that, or like a gene level expression information or a sequence that kind of models what you think it should. I think those are like I think those are something that we hope to work up to. And I know that there's already a lot of work being put into Eric Schmidt thing. I suspect probably those guys are probably doing a lot of that. So I think our focus is like sort of build up these like basic capabilities, and then but yeah, eventually to be able to generate like either gene level or for training kinds of things, or sequence level for maybe even designing genes for like specific purposes according to like whatever the scientific literature says you should do. So yeah, definitely not from this, I don't think, but I think it's possible. Yeah. Yeah. Yeah,
Cool.
no, and the other really interesting thing that, and this will be the next thing that we try that I should mention, is that this is actually right now it's trained. This technique actually probably we have to test this, but there's like an order of magnitude more data on bulk, what's called bulk data, where you, like I said in the beginning, you kind of like you have a sample that's reasonably large piece of tissue, and it has lots and lots of different types of cells, and you get the gene level expression data on that whole sample all mixed up together. And so that there's actually an opportunity here to apply exactly the same technique to that and use it for like the classification of that sample, like say what were the changes, either classification or other things, where like what was the change in gene expression between when I gave this drug, or for a diagnosis, like is this cell like exhibit this, or this sample exhibit this gene, or this exhibit a disease because the RNA is being expressed to counteract the disease or whatever. So so there's like a really cool opportunity which is what we're probably going to start working on like later this week or something, or next week. And so maybe in the whatchamacallit, the AI in action, I'll try to, if we have some cool results, then I'll try to sort of do an AI in action on it.
Awesome.
Cool. All right. Well, it seems like I've kind of run out of steam. Is there, so I'm happy to talk more if people have questions, but I want to give other people an opportunity to go over stuff too.
Yeah, well, I guess we'll open the floor to like any other papers that people want to explore. I had in my mind like clearing up the voting list that we had on Slido, where like obviously some stuff gets stale, and we might want to just refresh it and make a new Slido.
Yeah.
Like, what do people want to cover, or what have people read recently? Open up the floor. Mm,
Did
any-
Modern Bert come with the actual paper, or was it just like a blog post?
Yes, it did. Modern Bird, very, very full-fledged. I was actually literally just adding Modern Bird to the AI engineering reading list, like right before this.
Nice.
Yeah, I would love to honestly just get hands-on with it. Like, yeah, it seems like a good paper. What do you like about it? Oh, Sebastian says cooperation among LLM agents. What is this?
It's a paper that's suggesting like a benchmark for evaluating like a organization of LLM agents.
Hmm.
Uh, yeah, yeah. I also want to hear about Modern BERT too, but I don't know if anybody's ready to present that.
Yeah, I don't think we're ready. We don't have to do it today. We can do it next week. But yeah, I mean, I guess I guess I'd be down for a Modern Bird slash LLM agent cooperation thing next week.
Oh, I was kind of. I don't think I was too interested in like going over cultural evolution of cooperation. I just wanted to like have like if we don't have anything else to cover, I'll just run through it quickly.
Sure. Yeah. No, I mean, nothing for today. I was gonna end it early, but if you like, I'm still trying to understand this donor game that's on the paper. But if you already have it in your head, then yeah, by all means.
Yeah, I can go through it. So let me.
So this is related to a paper that I've been talking about recently with multi-agent steganography, where they're hiding messages. It's a kind of game, but more sort of safety oriented.
Oh shit.
You got it?
Hi. Yeah. So I'm just. Where is it? Okay, found it. Text PDF. Hopefully this. Yeah, nice. Okay, I'll share my screen.
There you go.
I'm not. Yeah. I also have a Jupyter notebook, but I'm not gonna open that up. Instead, I'm just gonna open up the GitHub repo for it. This is my own GitHub repo. I don't think they actually published a GitHub repo, but okay. So in general, like to like set the base of this paper, they're trying to explore like how agents are in the hypothetical scenario that you're building an organization that consists of multiple agents interacting with each other. You'd want these agents to pursue like the collective interest of all the agents to put together instead of a single agent just looking out for itself and trying to maximize rewards for itself. And they posit that by having um, they posit that like the game of the donors game is a representation of like collective benefit of an organization. And so what the donors game is a game that is covered in game theory, which suggests I'll just kind of draw it out. So you have like three players. You have player one, you have player two, you have player three, and there's the idea is that if the whole of the three players will benefit from donations between each of the agents. So one donated three, three donated two, back and forth. And then over the course of like at the limit of like X amount of donations, if there's a lot of donations between the different agents, one will end up with more like sum of money than had none of them donated in the first place.
Yeah.
And a key principle of this is that it has to be a positive sum game, meaning that there is an increase in like the total pool of wealth / resources in the collective in the organization. So what they do in the paper is that whenever one donates to three, let's say one donates a two quantity of something, then that gets multiplied by two when three receives it. So three ends up receiving four, even though one only lost two. So this is just a representation of positive sum. And so if you just continue cycling this over and over and over again, assuming that everybody's like completely everybody's just continues to donate to each other and nobody just hoards the information and decides to just keep the money, then you eventually have like three with assuming that 10 start one starts with 10, assuming that everybody starts with 10, he loses two, which leads to three. So he goes from 10, 3 goes from 10 to 14, and then 3 donates also 2 to the second agent, and then 2 gets 14. 2 goes from 10 to 14, and then 2 donates 2 back to 10, reciprocating the original the original donation, resulting in 10, which went from 10 to 8, going to basically 12, right? So that's the idea behind the donors game. Like if everybody donates in this system, then every single player is going to result in having a greater sum than if they just didn't donate at all. And so that is what they're basically trying to illustrate with agents, and trying to see like which they explore which models are perform best in this game. And they end up finding, I think, that and here's a design, but just to not get sidetracked. They point out that, okay, here's the results of the models. Cloud 3.5 Sonnet performs the best, and something that was interesting is that GPT-4o didn't perform very well. And as far as what I gathered from the paper is that GPT-4o was like too careful with its donations. Like GPT-4o was would refuse to donate. Was like too skeptical of donating whenever it would be his turn. So yeah, he just wouldn't donate. Cloud 3.5 Sonnet understood the premise of the game, and so just was more liberal with donations. And again, something I didn't point out is that if, for example, 3 just decides to keep all the money, then this game completely just falls apart. 1 at the worst case will just end up losing money, and then 2 won't get anything, and then maybe 2 donates to 1, and so the ultimate like loser is going to be 2. So that's what you got to take into account. So 1 is that it's a positive sum. 2, that you can actually see the reputation of the players. So you can see, for example, if 3 donated to 2 or 3 decided not to donate to 2, and based off that information, you inform whether or not you should actually carry on donating. So that is the idea of the paper. And I didn't prepare to present this, but I just want to have kind of the crowd's take on whether you guys think this is like an accurate, a good benchmark to judge models on, for basically organization-wide agent clusters. Well, just any thoughts on that?
Any thoughts on the whole thing?
The donors game as a benchmark, basically, for
I like
donors
it.
game as a couple of questions. Donors game as a benchmark for like agent organizations, and 2 is what is 2? Oh yeah, agent organizations in general.
Yeah, I like it. I want to let other people respond before I jump in. But anyone have takes on this? It's a nice paper. I can't believe I haven't heard of it.
Oh yeah.
I would like to see some like more models. I think the whole like PvP 3-3 thing has been tried in crypto, and so I've seen it like play out, and I think more often than not it ends up with like the whole like somebody, it's like a game of chicken, right? Like whoever pulls out first keeps the money first, like wins essentially. But it's super interesting to me. But it would be cool to see other, like maybe some of the open source models, or like see how it plays out, because I do think as people like bring more agents online, or as agents become useful, this would be like an important thing to like figure out how to incentivize them all to play nice.
Yeah, you mentioned the game of chicken, and then they did kind of address that towards the. They tried to prevent people from gaming, prevent the agents from gaming the system. Somewhere in the paper, they discuss how they actually don't disclose when the round is going to end, because ideally, if you know when the round is going to end, you just wait till that round and just like keep everything. So that's a interesting just policy that they added. Oh yeah, and here were some of the results. I forgot to cover this, but some of the results of like me running this locally on GPT-4, and total money per round. This is basically just one of the metrics that they were using to determine the effectiveness of the paper. Well, the effective collaboration. Basically, this is like the overarching metric behind it. Like, if the more the organization donated, the more the the virtuous cycle would take over. And you can kind of see a trend where the generation one. Oh, and another thing is that over the course of so the idea is you start with like a set of agents, and I'm not sure if I'm sharing. Right?
Yeah, we
Okay.
can see.
Okay, so over the course of you start with they started with 12 agents, and what would happen is like you would run a donors game, a donors game iteration, and then towards the end of the donors game iteration, you would have the top 6, the top half, be able to descend. They're basically going to be the parents for the next generation. So you're going to give the original strategies of the top 6 performing onto the next generation, and so on and so forth. And so that's what these different G's represent in the legend. And so you can see some like upwards trend in like accumulated money. Then I did like average donation percentage.
Was this like an evolutionary algorithm, meaning like you
Yeah.
combine, so like when the next generation is some sort of combination of the strategies of the previous generation?
Yes, exactly. That's what it is.
I see.
And you can actually see the prompts. I love what they did with the way they illustrated the prompts. It was very clear. So in the where's the evolution prompt? Okay, so the system prompt, that one's passed. This one just like describes the game. Donation prompt. This just determines how you should donate, and it also includes like the reputation trace of who you're donating to. And this was also something that was interesting, how they did the donation trace. And then the strategy prompt. Yeah, okay, so this is basically what you're looking at for generation. If it's a first generation, it just generates some strategy. If it is any subsequent generation, it is going to just inherit the top strategies of the previous game. So as described here.
Oh, okay. So it's basically, that's really cool. That's using the LLM to kind of merge this or like sort of combine strategies from the previous generation. That's
Exactly.
interesting. Nice.
Yeah, and I'm thinking of expanding this to like be more direct, like a lineage where the top donator is gonna have like 3 children, the second top donator has 2 children, the third top donator has 1 children, just like replicate what is it called? Natural selection.
So I don't really understand, like, what is so I was just poking around online. I don't understand the difference between the donation game and Prisoner's Dilemma, except that I read that the donation game is a version in which you have it's a positive sum, which is what you said as well. So why does that matter? Like, what's the distinction here between Prisoner's Dilemma and the donation game?
Remind me what the Prisoner's Dilemma is about.
Oh, okay. It's sort of similar in which if you think about 2 prisoners being played against each other to by the DA, right? Then the DA is like, oh well, like if you don't sell the other guy out, then he's gonna sell you out, and you're gonna go to jail for like 5 years. But if you if you each sell each other out, then each of you gets 3. If you sell the other person out and they don't sell you out,
Yeah.
and if neither sells each other out, then you only get 1, right? So like there's been a lot, it's been probably the most studied game. And if you and so there's like this iterated Prisoner's Dilemma, which is like this where you go over, if you play over and over again for an infinite amount of time, then there are strategies for which you can induce cooperation by your opponent, right? and so like, yeah, super well studied game. And this is, I think, related game, but I guess since you don't understand, you might have trouble distinguishing the two since you haven't encountered it before.
I think it. I think they both represent like the similar dynamic where you want what's beneficial for like you to like as the sum of all parties involved, rather than
Yeah.
the reward for a single one. I'm actually you could potentially do the same thing for Prisoner's Dilemma. I just don't know how the information transfers between prisoners in the prisoners game.
I think that the setup, it sounds to me like the setup is the same. It's just that you have a growing, like you have this positive sum game, right? So that's and that changes the dynamics, but I don't understand how. So okay, yeah. Anyway, maybe I'll look into that and put it in the chat later.
Yeah. Oh yeah, and just gonna go through the rest. I don't think this graphic was actually very interesting. Oh, what happened here is on the third generation, some of the LLMs and instance and agent, and bear in mind this is what was this for? Like the run that I did was, so it's 4 generations, 6 rounds for each generation, and 4 players. And one of the players on the reason you see like a big spike here is that one of the players here decided to just donate more than all of his money. I basically told them to do like a percentage between 0 and 1, and they just decided to do 1.3. So I just donated everything, which led to a massive spike in like the overall game pool here. Then
LLMs are terrible in numbers. Yeah.
yeah. Yeah, it would be nice if you could do like constraint generation, constraint decoding within like the boundaries of like something to something. That would have been better, but it is what it is. And then maximum wallet amount per generations, and this is basically the lucky guy who received the entire wallet from somebody else. But yeah, I'm probably gonna do another run of this with that actually is like a perfect that is in line with what they did on the paper, which was like 12 generations, or like 12 rounds each generation for 6 generations with 12 players, something like that. Yeah.
I'm curious. Have you shared this with the authors, being that they didn't produce any code for the paper?
I did. They liked my Twitter post, and that's all I got.
Yeah. Yeah. So this is fire. The fact that you put it in code is fire. That's really cool to see it in code.
Yeah, it's pretty easy to run too. Like it's just a Jupyter notebook. And it's basically completely enclosed since it's just like a simulated environment. So you can go ahead and try it yourself. So it's just this. Yeah. Yep. So that's all I have.
Can you share that notebook or GitHub or whatever?
Okay, I did not. It's here.
Yeah, the tweet as well too, please. Retweet or something.
The what? Oh, the tweet.
Yeah. I'll retweet it.
Yeah, I'll take it.
Wow. Why is that a long thing? Cool.
Yeah, I don't. Oh, okay. The description was like AI generated, and it just wrote like 3 sentences.
That was a super nice. It's rare to have a re-implementation of a paper. That's always hard. Awesome. Great timing. Yeah, no comments. Like, Claude is very smart. I don't know what to say. The classic, like, you should try it on 01, right?
So, oh, what? What?
You should try it on 01 because obviously, this is a test of intelligence, right?
Yeah, I'll take funding for that though. Yeah, it's gonna get expensive. But yeah, for sure.
That expensive. So like maybe like
Although it does have chain of thought. Like it is not like purely O1. And I can kind of show that in the code. It does have it does think through whether or not it should donate, for example. And this is right at the beginning. Wait, where do I have this? Yeah, so here thoughts. This is just basically chain of thought.
Yeah, maybe running this on something like QWQ or DeepSeek will be like less expensive than O1, but like still interesting results. I have to try and run it.
Yeah. Yeah, I was thinking of using open source. But it actually wasn't too expensive with 4.0 mini.
Nice. I have no clue what the implications mean that 4.0 like was super stingy, but that's crazy. That's crazy to see.
What?
I said I have no clue what that means, what that implies that 4.0 was the stingiest of the models, right? Like OpenAI's training on the models.
Yeah, no idea.
Did you use 4.0 or 4.0 mini?
I use 4.0 mini.
Oh, I see. So maybe there's a model class thing there too, right? Because I think the comparison to 4.0 would be Haiku.
yeah. Not like for like.
Oh, that is interesting, right? Because they didn't. Wait, is that true? 4.0 is equivalent to Haiku?
I mean, I don't know if it's equivalent, but I think they're similar class. They're like in the same cost class. Let's put it that way.
So you're saying there's not a fair comparison?
No. You said you use 4.0 mini.
Yeah.
That is noticeably different than 4.0.
I haven't done that analysis. I wouldn't be able to tell you.
Yeah.
Oh, okay. So the fact that it's like growing like the pot of money is growing over time. I'm not sure if my graphic is a good comparison to this, if that's what you're thinking.
No. We're just talking about model choice.
Okay.
You said you use mini, so
Yeah.
yeah. Okay. Well, very, very cool. That was actually like very productive. Really well done.
Thank you.
I guess we'll do Modern Bird next week.
Yes, that'll be sick.
Yeah, Modern Bert would be awesome.
Awesome. Okay. Well, Happy New Year to everyone. We have continued our unbroken streak of paper clubs. This is all very hard to keep up, but we're through it. We're through the tough part.
Oh yeah, and finally I posted the X post in there.
I see it. All right.
Awesome.
See y'all.
Cool. See you guys. Happy New Year.
New Year.
Okay, bye. Happy New Year.